Today's topic is viruses. So this is the virus lecture. You're going to want to take out a blank piece of paper and write your notes on there as I instruct you. First of all, viruses that we're talking about today are not computer viruses. They're not social network viruses. And let's be honest, I don't know what half of these little symbols stand for. Today we're talking about the kind of viruses that make you sick. So they're pathogens or disease-causing agents. And we're going to talk later about what a retrovirus is. That's my idea of a funny science joke. Please take your blank piece of paper and fill it out in the following manner. What that means is write the words "what is a virus" at the top, and then leave yourself 2 additional lines to write that information in. And then "are they alive?" Leave 5 lines, yada, yada, yada. So please stop the presentation until you have recorded that information. All right, where it says what is a virus, please write it's a disease-causing agent with these 2 qualities in common. All viruses will have a capsid, C-A-P-S-I-D, fancy word for outside protein coat. So you see that kind of in the green. And then the second thing they have in common is nucleic acid. And you remember from chapter 3 that that means they have DNA or RNA as their nucleic material. Other than that, the shape and some of the other special features they have may be quite varied. Next, make yourself a T-chart that kind of looks like this. And then under "are they alive," put reasons for yes and reasons for no. Under reasons for yes, we're going to have these 2. They have a genetic code, again, either DNA or RNA. So write "have DNA." And secondly, like living things, they change over time or evolve. So they undergo evolution. Now you may not recognize this chart, but this was from, I believe, chapter 1, and it listed the characteristics that living things would have. Viruses are kind of in a middle ground. They have some characteristics of living things, but not all of them, and so we consider them to be non-living. The no ideas is that they cannot process their own energy. So they have to get ATP from some other cell. They don't have a mechanism for doing that themselves. And the key one is they cannot reproduce unless they are within another host cell. So if I had a bunch of viruses and I spilled them on a table that was completely sterile and remained that way, they would be unable to reproduce because they have no host cell to reproduce within. And they don't grow or develop. So just like a cat starts as a fertilized egg and it's born as a kitten and it develops into a cat, a virus is just the way it is from the very beginning to the end. So, are they alive or not? And this point again we consider no, basically because they cannot reproduce unless they're within another cell. All right, I think the next topic was what is their size, and I wanted you to have room for 2 things, or basically lines to write this, that bacteria are small and viruses are much, much smaller. That's the first thing. Secondly, write down bacteriophage. A bacteriophage is a virus that is so small that it can actually invade a bacteria. So this picture is just trying to show you some rough sizes. This is a red blood cell. You have millions of these flowing around in your blood. And by comparison, this is the size of an E. coli bacteria that might be living in your large intestine. Okay? These then would be our little virus particles. And if we now look at the E. coli and blow that up, again, now we can see teeny little viruses. And when we really blow up the E. coli, now you can see some of the detail, the different sizes and shapes that the virus particles are. This refers to nm, nanometers. A nanometer is 1/1000th of a meter. So as you can imagine, these are quite small. Next, we're going to talk about virus reproduction. And again, they do reproduce, but they have to be in a host cell to do that. And so I'm going to have you write lytic cycle, Lytic, and then the following numbers. Please write the number 1 and write it attaches to the cell. So this is a bacteria, and this is the bacteriophage, or the virus that's going to invade it. So please write number 1 and then write again, it attaches to the cell. And it wouldn't have to be a bacteria cell. It could be one of your cells. Step 2 is over here. Step 2 is that it injects its DNA into the cell. So you can see this little purple, that is the DNA of the virus, and it's now injecting that into the host cell. In step 3, the virus takes over and starts making more virus particles. So it uses the bacteria, or again your cells, mitochondria, Golgi body, to make the things that the virus needs. So we see in kind of these two pictures, it making more virus particles and then putting them together. So that's all step 3. Use the cell to make more virus particles. And then step 4 is where the host cell bursts, and now the virus particles are free to invade more cells. So step 4, burst and infect more cells. So our one virus particle has become 7. If that happened again, our 7 would become 49, and then our 49 would become a really big number. The point of that's the incubation period. The time between when you get the first virus invading until there's enough virus particles to make the person feel sick. So lytic cycle results in the bursting of the cell to release virus particles to attack more cells. The lysogenic cycle kind of starts the same, but then it goes a different direction. So in step 1 again, same as above, it attaches to the cell. Step 2 is also the same. It injects its DNA into the host cell. Step 3 is going to be over here. Okay? And that step is going to be the virus DNA becomes a part of the cell's DNA. So you can see the bluish color here. That was the host cell's original DNA, and the purple is the virus DNA. The two are now one piece, so that in step 4, every time this cell now divides into 2, it copies not only its DNA, but the virus DNA as well. So please put for step 4, copies virus DNA when the cell divides. Here, I'll put a 3 here in case that makes you unhappy that there was no 3. This is kind of a dormant side of the cycle, I guess. So the cells in your body are making more and more copies of the virus DNA, but you're not going to be sick. What happens, though, is something in the body triggers these cells to separate out the virus DNA and basically start making virus particles. So for step 5 here, simply write goes lytic. And then draw a line up to number 3 in your lytic cycle. As you remember, that's where the virus now uses the cell to basically make more protein or make more virus particles that are going to burst out of here and infect more cells. Here's a couple examples of lysogenic viruses. So herpes simplex, whether you get it as a genital infection or a cold sore, it goes through kind of cycles where you see the infection, and the person would have symptoms, and then it goes away. You don't want to kiss anyone with an active cold sore because that will then get the virus potentially into your body. Shingles is a disease older people get, and it is when the chickenpox viruses reemerges as a disease kind of the skin and the nervous system. And then HIV/AIDS is probably the one that we want you to know. So you can have HIV and you have the infection. The virus is growing in your body, but it's still in those cells and it's not causing active AIDS to occur. The next page here is just a bunch of different diseases. These are bacterial, and you'll notice that we know the pathogens that cause them. Scientific names, capital letter here, lower letter here, and then written in italics, and some ways of preventing those. You don't need to write anything down from this slide. I will not ask you on a test what the specific name or pathogen is for specific bacterial infections. These are some virus ones. And you'll see that we talked about having either DNA or RNA as the nucleic acid, and some diseases that they're connected with. A retrovirus, and write this down, simply means that it has RNA as its nucleic acid. As you remember from our lessons on protein synthesis, RNA only has one side to it. So when it makes copies of itself, since there's not another side to kind of double check that it's right, the RNA will mutate or have mistakes more often, and therefore it evolves faster, and it's sometimes hard for us to make medicines to treat it because the virus keeps changing. This slide just has a couple other causes of diseases. So infectious diseases can be bacterial, viral, fungal, or protists. And funguses are just things basically growing on the outside kind of your skin. I guess that's not always true. Athlete's feet is a fungal infection. It's not specific to athletes. You simply pick it up at the gym on the dirty floor in the shower, and thus it got its name. Some protists or one-celled organisms cause disease. Giardia is something you can catch in the boundary waters by drinking the water. And then after a few days, the protists multiply and you get symptoms. To avoid this, one should either use iodine to sterilize their water, boil the water, or buy a filtering system that would filter the Giardia bacteria out of the water. All right. The symptoms of the common cold, if treated vigorously, go away in 7 days. If left alone, they will disappear over the course of a week. Why then, if it doesn't make any difference, do we take cold medication? Basically, cold medication helps to control symptoms that make you feel icky and unhappy and kind of dull, but it does nothing to make your cold go away any faster. How do I know if I should go to the doctor? Well, basically, if you have a viral infection, you're going to have a whole bunch of symptoms. Since this is viral, antibiotics will not help to make it go away. So there's not a real point in going to your doctor because, again, there's not an antibiotic that's really going to help you. In terms of bacteria, the key thing is often you only have one symptom. Ooh, my ears really hurt, or my throat really scratches. So in that case, maybe you do want to go to a doctor because an antibiotic may help. Very important on the test, write down, antibiotics will treat bacterial infections, but do nothing to help a viral infection. Last point, interferon is something that helps your body fight a viral infection. Basically, it's a protein produced by an infected cell and released to neighboring cells. It says, hey, neighboring cells, there's a virus. As the neighboring cells take it in, it changes the permeability of their cell membrane to help them better, at least slightly resist the viral infection. That, with 2 seconds to spare, is our virus lecture. Thank you.